But he had difficulties in his flight to Heathrow was delayed this morning. So pleased that he's managed to drive all the way over from Heathrow, and he's going to talk to us about feedback, lineages, and cancer therapy. John.
Yes, thanks, Charlie. Thanks for inviting me, and it's a pleasure to finally be here. This is joint work with a bunch of different people. Probably I'll only be able to talk about the work with the first three, Arthur Lander, who's in developmental cell biology at Irvine, a former student, Hamad Yousefpour, and a former postdoc, Shangrong Li. And so basically I'm going to talk about at least one set of models that we have for cancer, and basically it's a tumor modeling and cancer modeling is a multi-scale problem involving processes on the microscale involving proteins, DNA, RNA. They're interacting through signaling networks, which tell cells what to do, which deals with the tissues in the whole organism, and then of course it's affected by the environment. And then this is kind of the more global picture. On the more local picture, you have tumor cells interacting with the microenvironment, and all of these factors are affecting how the tumor cells are interacting. And so what you really want to do with mathematical modeling is mechanistically connect the scales to ultimately basically be able to predict both the progression of tumors and its outcome. So cancer is basically the loss of control. And so here's an example, just a cartoon of basically how a tumor can grow. If you have a simple epithelium where you have one cell, basically epithelial cells, the one cell thick layer that are dividing, basically these are, they divide inside from outside. So they're bound by a basement membrane above a stroma where there's blood vessels, and inside here would be a lumen. So this would be, for example, a piece of colon. Then, as a result of changes, which could be genetic or epigenetic, the cells could then start to proliferate and so produce an avascular tumor that would basically give a local increase in number of cells. And then eventually what can happen is that the cells can break down the basement membrane and invade the stroma where you have the blood vessels to form a vascularized tumor, and at this stage is when the cancer can potentially become metastatic. And so this loss of control is basically has been thought to, well, of course, since you have accumulation of cells, it's really the cell number that's being lost, and that typically is tightly regulated. Tissues know how many cells they should have, and there's a balance between birth and death. And so one of the traditional views is that cancer is a condition of the cell cycle, so dysregulation of these birth-death processes, so you have more birth than death. And it could be done by, for example, acceleration of the cell cycle, and that's an example of direct control. But there are also examples of indirect control, and in fact in normal tissue, homeostasis and growth are tightly regulated by feedback control. And so here's an example of a lineage, so the fundamental component of a tissue, where you have stem cells, some set of progenitor cells, and then a terminally differentiated cell. And typically what happens is that the more differentiated cells produce factors that negatively regulate the various properties of the less differentiated cells. So for example, here these are these V's denote division rates of cells. So the differentiated cells could produce factors that reduce division rates, and that's a form of control. So you could think mathematically that's a simple way of modeling that would be like logistic growth. But it turns out that an analysis shows that that's not a very robust mechanism for maintaining homeostasis or controlling growth. Instead, what's a more effective control mechanism is to control what's called the self-renewal probabilities. So the fraction of cells that remain at the same lineage stage rather than differentiating. And so these cells can also produce factors that negatively regulate these self-renewal probabilities. So those are the P's. And that produces a very robust control strategy. And so, and now there's been a lot of examples shown that in fact there's the factors which tend to be from what's called the transforming growth factor beta superfamily. So a whole bunch of proteins that do this that have been shown to affect exactly these parameters, the self-renewal probabilities. And they can be positive effects and negative effects. And so this feedback on the self-renewal fraction basically says that the behavior of the cells, so whether they're acting like stem cells, so they're self-renewing perpetually, or in cancer, that's basically the definition of a cancer stem cell, or a committed progenitor cell, which you would think would die out after a certain number of divisions, but that may not be intrinsic properties. They may be a response to feedback and feedback control. And so now, how about in cancer? Well, in cancer, there's evidence that lineage progression occurs as well. And so here's an example where, as I mentioned, so these cancer stem cells are the ones that have potential for indefinite renewal. And so here's a collection, basically a breast cancer, there where the cancer stem cells are marked by well, these are two different surface proteins, and the cancer stem cells are the ones in this region, so roughly there's about 5%. So what was done is the tumor was grown, extracted, cell sorted, and about 5% of the cells have these stem cell characteristics. Then if you sort for these cells, so you create a collection of cells that's basically 99% of these stem cells, you reimplant it in another mouse, you let it grow, will produce a tumor, extract the cells, you get about 5% again, even though you started with 90%, 99%. And so there is even in cancer, which, for which there's a lot of things going wrong, this fraction is quite robust, indicating that there's some type of feedback going on, at least suggesting. But you can also manipulate the numbers. And so one way of manipulating the number is by doing therapy. And so, for example, chemotherapy, so this is another type of breast tumor. And so this is the size as a function of days after treatment. And again, these are the same markers. And so going from, again, about 5 or 6% of the cells, after doing secondary transplant, so you basically treat, transplant, you can increase the fraction of cells up to 22% or more. So you can dramatically increase the number of stem cells, and stem cells are very robust to treatment. So in fact, this is one reason why it's thought that you can acquire resistance, is that basically by treating, you're killing off the less resistant cells, you're leaving the more resistant cells, and those are the stem cells, and those are exactly the ones that can recreate the tumor. And so you're selecting for resistant cells, and so this is a source of resistance. And so, of course, from the mathematical point of view, it's a very challenging problem, as I mentioned, because you have this linking to scales. And so one wants to build models that bridge the scales, and I probably won't have time to talk about that, but I can talk about that after if anybody is interested, what we've been doing with that. But another key thing is trying to access data. And there's a lot of data, of course, that's out there, but not all of it is useful for mathematical modeling, and that's a big challenge. Just to go over a quick overview of the types of approaches, there's discrete approaches, continuum models, and hybrid models. So discrete, basically, you're tracking individual cells. Continuum, you're tracking volume fractions rather than individual cells. And of course, the advantage of discrete is that you can basically describe a lot of biological detail, With the continuum, you can go to much, but you're limited in scales because because of computational expense. But for continuum, you can do larger scales, but you can't describe transitions. For example, epithelial to mesenchymal transition, where cells going from behaving collectively, like epithelial tissues, to individually, like mesenchymal cells. And this transition is thought to precede metastasis. And then hybrid models, where you can combine both descriptions. And in principle, these should be the best because you could use the best features. So I'm going to focus on a continuum model to start, and then, but as I said, I probably won't have time to talk about the connection with discrete. But basically what we can do is introduce volume fractions for the different cell types. So viable cells, dead cells, and total tumor. We can have host cells and water cells. And then you just assume there's no void, so the sum of the volume fractions is 1. And then you can write down mass balance equations for the volume fractions. So Ui is the cell velocity, and Ji are the fluxes. Then Si are the source terms. And if one assumes for simplicity that the water fraction is constant and the solid fraction is constant, then you can decouple the water and the solid, and I'll just describe what happens for the solid. You can show in an appropriate limit. This will give you a free boundary problem. But in order to close the system, I have to tell you what the velocities are and what the fluxes are, and of course what the source terms are. And so I'm just going to describe the simplest model of soft tissue mechanics, where basically you're treating the cells like a fluid, but even a simple fluid, a Darcy's fluid. So basically the velocity is proportional to the force. And so the only type of stress we'll involve is a hydrostatic stress, but we can also have additional stress or additional forces from cell-cell adhesion. And so we'll assume that the cells are tightly packed so that they all move with a mass average velocity. And so then basically the velocity proportional to the force, which consists of a pressure force and additional forces, from adhesion. And then for the fluxes, we can use a generalized Fick's law. So we can relate the flux to the gradient of a chemical potential. And that chemical potential we can use here as being related to the cell-cell adhesion by introducing adhesive interactions, or basically just interactions between cell types. If one localizes, so there's certainly evidence for this. For example, here's a mix of neural cells and epithelial cells, and you can see they phase separate. And so we can incorporate this into a classic Cahn-Hilliard type approach, where we can think of the tumor cells as trying to separate from or at least be distinct from the host. And that's not always the case. And one can vary that by the choice of what this potential is. So if one has a double well potential here, you can enforce separation, but you can also take a single well, and that will allow mixing. And that's just a reflection of the interaction between the cells. And then, of course, the cells have to have nutrients to proliferate, and so we can just take a diffusion equation for nutrients, where they're provided by vascular system, and they're uptaken by the various cell species. And it's quasi-static because basically on the cells proliferate on the order of a day, whereas these guys diffuse on the order of minutes. And now here comes the lineage point of view, so that's all generic. But now the lineage is that you have stem cells, committed progenitor cells or transient amplifying cells, terminally differentiated cells, and then also dead cells. And so basically the cells, while they need nutrients, they divide with rate lambda, and then P0 is this probability of self-renewal. And so this term is combining the fact that P0 of these guys stay at the same stage, and 1 minus P0 differentiate. So you get 2P0 minus 1. And then the cells can die by various means, either at a particular rate of death or a rate of death that depends on nutrients. then the differentiated cells here produce 2 differentiated offspring, so you get the 2 here. And then these guys can self-renew to some degree, and then you have death. And then these differentiated cells produce 2 terminally differentiated cells. These may divide a limited number of times, and then they may also die. And the dead cells source basically is just the sum of all of these guys, but then there's a rate of clearance. Okay, and then now the negative feedback factor, well, that's going to be produced by various types of cells, the differentiated cells mainly, so that's where what we'll assume. Then it can be uptaken by various cells that will diffuse and decay. And then how that affects things is that it changes the self-renewal fraction by basically decreasing it. This is just a simple Hill function type decrease, where you have a gain with respect to this factor. And so if you just include this into the model and you solve the equations, you start with something uniform, then you'll get a tumor that basically contains uniform distribution of cells. But that's not everything, because in addition, that was only negative feedback, but in addition you can have positive feedback. And so one example of positive feedback is Wnt, and it's known that this provides positive feedback for self-renewal of stem cells. It's produced by stem cells, and there's an inhibitor, a couple of different inhibitors, DKK or SFRPs. And if you have activator, inhibitor, well, that suggests a Turing model. And in fact, so when you have a Turing model, you expect patterns. You expect heterogeneity. And so what evidence is there for that? Well, here's an example of, this is a colon cancer, and the green are indicating cells that have high Wnt activity. And even though Wnt is supposed to be constitutively activated in colon cancer, you see actually there's a heterogeneous distribution. And there's a recent paper that shows, in fact, that Wnt and DKK can both be produced by stem cells. And so that's in skin. I don't know anybody who's tested the same thing in colon, but I suspect that something like that occurs as well. So one can then model this, well, just as simple a generic type of model with a Geiger-Meinhardt system, where now you're modeling the Wnt concentration. There is a nonlinear production term. There could be a background production term. And so this nonlinear production term is motivated by the fact that, well, there's multiple ways to upregulate Wnt. So Wnt has a whole cascade of effects that can give self-upregulation, but it's inhibited by the inhibitor. And then likewise, there's production of the inhibitor, and then decay. And so now how this affects the self-renewal fraction is that you have positive feedback from the Wnt and negative feedback from the growth factor, the TGF-beta guy. And so this is one particular way of writing it. There are many ways to write it. It's a simple way to do it. And of course, the system is complex, so you need efficient solver. It's actually a fourth-order system because of the adhesion. But let me show what happens now. And so if we put this together, let me show you the stem cells first. So what happens, instead of the uniform state, what you instead get are clusters of cell. So these are high stem cells. Concentration, so you it doesn't look like anything's happening, but it actually is starting to finger. If I show you the total tumor, you can see that, and then let me show you everything you can see in this terminally differentiated cell. So this is the stem cells clusters here. You see they're right at the boundary. They're driving fingering, and and then you can produce additional basically hotspots or niches for the stem cell, and you get a highly invasive fragmented tumor. And so if you analyze that, so here's your free boundary problem. You have the tumor boundary. You have these clusters of stem cells inside, and then this is the Wnt. And so what we were assuming was that the Wnt production depends on the fact that cells have some nutrients, and so that biases it a little bit instead of being right at centered where the stem cell clusters are, the Wnt concentration is centered a little bit more towards the boundary. So you have higher self-renewal towards the boundary and less self-renewal in the back, and that drives the fingering, and that drives the instability. You can get the same in 3D. So these red are the stem cell clusters, and they're driving invasive fingers, and you're seeing the back, and you can get increased numbers of them basically because you have small concentrations of stem cells that get into the right environment, and that creates a niche for them. And if one varies now the feedback gain, so the negative feedback gain, you can affect both the size and morphology of the system. So this is a plot of total volume versus time. And if you don't have any feedback, you get this highly invasive tumor. As you start to this is negative feedback, so as you start to increase the negative feedback strength, you reduce the clusters, you reduce the volume, and you can eventually get something that's nearly steady, but not completely. Now, that's the complicated model. Question is there any experimental evidence for it? Well, there are evidence for each piece of it, but how about when you put everything together? And so, as a first place to look, we can actually look at what are called mini-guts or colon organoids. And so there was a review paper in Science last year talking about morphogenesis. And one of the articles in there was about producing these colon organoids from a single stem cell. And in this paper, they were talking about human, but these guys, basically Hans Klever's group, did it a little bit earlier with a mouse. And the similar results. The key is kind of what you're mixing into the environment to get things to grow. And so starting from a single stem cell, here's their cartoon that basically what happens is you form this niche where you have a stem cell that's surrounded by its supporting cells, and that will eventually drive fingers, which are basically the buds and the crypts. It's trying to form a crypt, which in this case is coming out to be a bud. And so this is the cartoon, this is the experiment, and this is our simulation. And so qualitatively, the behavior is similar. You have these buds being driven by stem cells. So this green is again the Wnt high cells, or in this case, it's these indicating these stem cells. And so that's very analogous to what we predict. Now, a test of the model would be to say, suppose you change the conditions. Can you make the prediction? Can you see the similar change? Can you make a prediction about what should be seen in the experiment? And so, well, we have two things. We have Wnt and we have the inhibitor. And actually, we have the inhibitor, but we also have the negative feedback factor. So if we add Wnt to the environment, then what the model predicts is that if you don't do anything, this is your natural state, and this would be your, so this is what we can call our control. And that's analogous to what is seen in the experiments. Now, as you start to increase the Wnt concentration in the environment, what happens is that, well, you get larger clusters, and you get more stem cell clusters around the boundary. So larger organoids and more stem cell clusters around the boundary. And in fact, that's what you see experimentally. So this blue are, again, the stem cells. And then as you continue to increase the concentration, you get even more, and you get now, in the model, a more continuous distribution of stem cells around the boundary. And again, the shape, it's bigger, and the shape is more compact. And again, that's something similar to what is observed in the experiments. You can also change the negative regulator. So that was the positive factor. You can do the negative factor by adding, in this case, since it's colon, the TGF-beta member is BMP, bone morphogenic protein. And so if you don't do anything, this is the same guy I showed before, but that's just blown up a bit. And this is the control. This is from a mouse. And now if you increase the negative regulator, you drive it, you make it smaller and more compact. And again, that's what you see in the experiment. So the model is predicting at least the right trend for the experiments. And that was for, but interestingly, the model makes additional predictions, so that was just adding a bunch. What happens if you change it gradually? So we go from none to more, and so the picture I showed was this guy. But actually, if you start increasing the negative factor, what happens is that you actually make the shape more unstable. You can actually, so these are all at the same time, but with different amounts of the factor added. And so, in fact, you can make the tumor or organoid more invasive, or at least more branched, more fingered, by adding a little bit of this factor, and then you bring it back down when you add a lot of it. And this is actually well known in cancer, which is that in particular for these types of family members that they can behave differently under different circumstance. And here we show even just with this only the feedback mechanism that non-monotonic behavior can be captured. There may be many additional things that go on, but at least this is one. So the response, even though you look like you add it and you make it nice and stable, the path is not so simple. Okay, 5 minutes? Okay. So now you can say, well, so those were in normal tissues. What about cancer? And so in cancer, well, one thing you would like to do is figure out where the stem cells are. And so there was a nice reporter mechanism developed by Pajank at UCLA, where they found that basically cancer stem cells tend to have low proteasome activity. So they add something that would ordinarily be degraded if you had high proteasome activity. If it's low, it fluoresces, and so the green are the cancer stem cells. And this is a spheroid that they grew. So this is a spheroid they grew with 50 microns. This is actually about 250 microns across. It's about 50, so like 5 cells, and this is like 5 times that. And so what you can see, again, the green are the stem cells, and you see these clusters at the boundary. And it's kind of a pancake-like structure. It's not so easy to see, but you can see the clusters. It's not homogeneous, and the clusters are at the boundary. So now, so again, it's at least qualitatively similar to what the model's predicting. And so now what you can do is, since you have lineages, and so you have different cell types, you can imagine trying to design a therapy that takes that into account. So for traditional therapy, well, the problem is that you develop resistance. And so one of the mechanisms by developing resistance is that you can increase the fraction of stem cells. So basically that occurs because, one, the therapy is more efficient for the differentiated cells because they have less well-developed repair mechanisms. And in addition, so that means automatically you're not killing the resistant cells. And then second is that you relieve, by killing the differentiated cells, you relieve feedback. And so you've removed their feedback, you've killed off, so you're killing off the guys that are controlling them, and so you can dramatically increase the number of stem cells as a result of both of these mechanisms. This increases the fraction, and this increases the number. You can also imagine, well, you can have differentiation therapy, which is that you can try to add something that will drive differentiation. And people are trying this. You can do it through Wnt, you can do it through Notch, a bunch of different ways to do it. And one can then, but you don't really know how much to do. And so one of the ways you could try to do to deal with this issue is that, well, if you're able to if you can drive differentiation, then you can kill off the differentiated cells with this strategy. And so the idea is to try to combine them. And so here's an example of what happens. So this is a plot of volume over time for different types of therapies. So this blue curve is the case when you don't do anything. So the tumor actually will grow, then it gets under feedback control for a while, but then it will eventually take off and produce these basically fragments that invade. Now, if you're in the state where the tumor shape is not changing too much, so that's this guy, then you could try to say, well, let's apply radiotherapy. And so you apply radiotherapy, you cycle, and you kill off a lot of the tumor, and you reduce the number of these stem cell clusters. But as soon as you turn off the therapy, you get a rebound, and then it grows quickly again, producing these fragments. If you try differentiation therapy, so that's the red curve, you drop down the tumor size, you do a lot to get rid of the stem cell clusters, but as soon as you turn it off, it comes right back and it behaves just like you didn't do too much at all. But if you combine the therapies, that's this purple curve, you can actually drive it right down to 0. And that's very unusual with a continuum model. It will never come back. And so you take two therapies, neither works by itself, but together it works. And so the reason it's working is the following. This is the stem cell fraction as a function of time. And so again, the blue is when you don't do anything. The green is when you do radiotherapy. And so you're going from something that's about 13%, and you drive it way up to over 30% when you're cycling, because you've relieved feedback and you're selecting for those cells. And so as soon as you turn off therapy, you have this stem cell rich tumor that then happily grows and regenerates. When you do differentiation therapy, that's the red curve, you drive the stem cell fraction down, as you would expect, but now the system is under this external regulation, so when you turn it off, it comes right back and will recover. But when you combine the therapies, you drive the stem cell fraction all the way down to 0. And so that's one example, and it's kind of indicating the one potential strategy for dealing with this. This is just an experimental image to show that when you do a radiotherapy, you can actually, the green are stem cells compared to nothing, you dramatically increase the fraction, like what you predict here. But but that's just one example. So now you could say, how robust is it? And can you predict response? And so one thing we did was we used machine learning, basically logistic regression, to try to predict the outcome of therapy. So we tried a bunch of different therapies. Combination, differentiation, radiation differentiation, and so on. And then mixing the differentiation, radiation, and so on. Take a bunch of different shapes, produce a data pool, extract the features, and then try to guess or learn what are the important features for figuring out whether the tumor will respond and what's the best therapy. And so basically, we use a lot of description of the shape. We found the shape is as opposed to the growth path, the shape is the most important. But then the fractions of cells also can play a role. And so you can incorporate all of this into essentially parameters into the machine learning model, and then use all of this information that you gather from the tumor. And what we do is we use it right before you apply therapy to try to predict which therapy will work for that tumor. And so basically, in fact, what we found is that you get very good results even if your test set is your training set is much less than your test set. And so most of the time you actually use a big training set and a small test set, but we found this worked pretty well. So we tested with different environmental conditions. So if your environment is not producing anything, then you can get about 98% of the accuracy for these guys. And it varies. Interestingly, the low production, because of this non-monotonic response, is a bit harder to predict, because you actually increase the invasiveness when you add just a small amount. But when you get to the high case, you predict correctly. And so ultimately, the outcome depends mainly on the shape, not so much on the environmental factors, and not so much on the different cell types, but somewhat on the percent of stem cells. And just to give last one. So just to give an indication about what is working, this is a shape factor as a function of area. This is just two of the variables in there because there's many. You can see these blues are the ones that are effective. The combination of therapy is effective, and so it's effective actually when you have these a high shape factor, so meaning complex shape, but relatively small area. Okay, so see, I promised. And so what I introduced was a model for cell lineages with machine learning to predict outcome. There's a lot of other work that needs to be done. So with that, let me stop and take any questions.
Well, I have one question.
John, does this analysis apply or some portion of it apply to non-solid tumors, which are, the leukemias, the lymphomas, and so on?
Yes.
And some of this be
So that's
over.
right. So that's actually, in some sense, a little bit easier because those are not spatial, and so you can use the same principles. In fact, the same feedbacks still go through, but they're quite well described with ODE models, and so you can see exactly the same type of effects that I described here. You can have a crisis where your fractions go way up. Treatment will increase stem cell numbers for exactly the same reason as here. The difference with the solid tumor is that you now have spatial effects, and so when you have the spatial effect, then if cells get separated in space, that can relieve feedback just by itself because the factors can't reach them.
Do the oncologists listen to this type of material?
Well, some do, some don't. And so that's the hard part, but the Priyank at UCLA, although we haven't published together yet, we're moving towards that direction, and he, so that's a sample of one that is enthusiastic enough to give data, but the hard part is really getting data. And so I think that the principles are there. The question is more validation is needed. I've tried to get people to do this combination therapy, and I haven't yet succeeded, basically because it takes money to do that, and so I don't have the money to give them yet to do it. And then to get the grant, you need preliminary data, so we'll get there.
I think we'll move on to the next talk, and because there's a long session for questions afterwards. So can we thank John again?